We are iGEM Franconia, the iGEM team of the Friedrich-Alexander University Erlangen-Nuremberg, the Julius Maximilians University Würzburg, and the Technische Hochschule Georg Simon Ohm Nuremberg. iGEM means International Genetically Engineered Machine and is an international competition in the scope of synthetic biology. In this year's competition, we focus on a project helping to simplify the development of artificial muscles. We try to achieve this in two parts, by cross-linking different protein monomers to tissues that can contract like muscle. One way to have tissue contracted under controlled conditions is the usage of so-called molecular machines. These are a class of molecules that can deliberately change their conformation and their structure due to their nitrogen bond. For our purposes, we use an azo dye. Through light irradiation, the azo dyes can be excited to mechanically deform in their appearance. This deformation occurs during the conformational shift when the molecule's size is decreasing. To contract an entire tissue, it is necessary to use large amounts of these azo dyes. To connect these single azo dyes with each other in a tissue, the ends need to comprise additional conjunction sites like biotin. These binding sites can recognize their biotin acceptor counterpart that is also a part of the tissue. A biosynthetically produced tissue built out of catch-attack systems is essential for using those conjunction sites. Like a lock and a key, these components, catcher and tag, can be combined to one entity. Due to their structure, these units can be combined to long polymer chains. To bind the azo dyes to such a tissue, additional streptavidin proteins are attached between each catch-attack system. This enables each azo dye to bind two catcher modules, since they have a biotin molecule on both sides. Therefore, the single polymer chains can be linked through the azo dyes. To eventually connect these large constructs of catcher, streptavidin and azo dyes to a network, tags are vital. The connection of the single bricks to a catch-attack system, as well as the cross-linking of the molecular machines with the catcher molecules through streptavidin and biotin, induce the development of a large network. Through light irradiation, all bound azo dyes finally change their conformation and contract. This results in a change of the entire network. Our artificial muscle contracts. Another possibility to produce a contracting tissue is the fabrication of a dielectric elastomer actuator based on the function of a plate capacitor. Here, an elastic deformable substance, an elastomer, is embedded between two electrical conductive layers. The elastomer again is a catch-attack polymeric tissue. Carbon nanotubes build the conductive material. Applying voltage to the system, the Coulomb attraction to the carbon nanotubes puts pressure onto the elastomer, which leads to a two-dimensional expansion of the whole unit. The stacking of several of those units allows the contraction of even larger tissues, and again, muscle-like behavior can be observed. With both methods, the application of azo dyes as molecular machines and the production of a biological capacitor system, we try to fabricate artificial muscles based on biological systems. Our project for iGEM 2017, BEAM.